order for replication to occur in an organism, a pre-chromosome complex must first be assembled and is responsible for the recruitment of the chromosome complex.
This is accomplished in prokaryotes by the recognition of the OREC region, which is composed of multiple cis-acting elements to mark the site of where replication is to begin.
DNAA proteins binds to the DNAA protein binding sites, forming a large helical assembly, resulting in a melting of the DNA at the DNA unwinding element.
DNA B helicase is recruited to the single-stranded DNA by the help of DNA C and is used to further unwind the partially melted region, creating the replication fork.
The chromosome complex can now assemble and initiate the replication of DNA.
At the head of the replication fork is DNA B helicase that encircles the single-stranded DNA of the lagging strand. Its associate DNA G primase primes the single-stranded DNA with RNA primers in order for the DNA polymerase III to process replication in the 5' to 3' direction.
The clamp loader, which is composed of 7 subunits, 3 tau subunits, which are in orange, that serve as a tether to the core polymerase, which are green, and the beta clamp, which are pink, by a 24,000 C-terminal domain. Delta and delta prime are required for opening and closing the beta clamp around the single-stranded DNA proteins by its ATPase properties, as well as chi and psi subunits, which are purple, that are needed to add single-stranded binding proteins to the lagging strand.
The clamp loader recognizes the replication fork and recruits a beta clamp dimer to the prime template, forming the pre-initiation complex. Conformational changes to the beta clamp enable it to have higher affinity for the alpha subunit of the core polymerase. After core polymerase has been dimerized by the tau, then synthesis of DNA can begin.
Synthesis of DNA occurs in a semi-conservative manner, in which the leading strand of DNA is synthesized continuously, while the lagging strand is synthesized discontinuously, requiring multiple initiation events. Since DNA polymerase III replicates both template strands in only a 5' to 3' direction, then replication of the lagging strand must form a trombone loop that orients it into the same direction as the leading strand. This was proposed by Bruce Alberts.
Helicase anchors the chromosome to the lagging strand, enabling the primase to synthesize a new RNA primer before every Okazaki fragment. The clamp loader will load a new clamp and core polymerase to the prime lagging strand and proceed to synthesize the next Okazaki fragment.
Synthesis on the leading strand has a single initiation event. Primase synthesizes one primer on the leading strand, and core polymerase continuously synthesizes the new daughter strand.